My name is Michelle Mellion. I am the Executive Medical Director and Head of Neuromuscular Clinical Development at Fulcrum Therapeutics. These are my disclosures. Today, I will provide you with a brief background on Fulcrum, provide an overview of the losmapimod clinical development program for the treatment of FSHD, summarize the main results from our phase 2 clinical trials, and provide information on our planned next steps for clinical development, as well as provide information about our early access program. Fulcrum Therapeutics is a biotechnology company based in Cambridge, Massachusetts. Our goal is to find treatments that target the root cause of genetic diseases. We are working to advance research targeting severe diseases and conditions including FSHD, sickle cell disease, and other rare diseases. And in these efforts, we try to work in partnership with leaders from the patient advocacy and treatment communities to help build awareness and to support research that can lead to new treatments. I know that you are all aware that FSHD is a rare, serious, heterogeneous, and variably progressing debilitating disease. The root cause of FSHD has been identified as the aberrant expression of DUX4 in skeletal muscle. DUX4 expression leads to progressive muscle cell death and fat infiltration, resulting in muscle weakness and eventual loss of function. FSHD initially affects facial and scapular muscles, eventually progressing to the arms, trunk, and legs. The impact on people living with FSHD includes decreased ability to perform activities of daily living, loss of independence, including the ability to work, and significant disabilities, often requiring the use of a wheelchair. The primary treatment goal is slowing or stopping the loss of muscle function, but currently there are no approved therapeutics. DUX4 is required for development in early life. It is supposed to turn on and off very quickly. However, when DUX4 remains on beyond the time it is needed for early development, such as in FSHD, the result is the disruption of normal cell function and the triggering of muscle cell damage. We and others have shown that P38 plays a role in turning on and off DUX4. What we are showing on the right is a cartoon of a cell with P38 activity. Losmapimod works by decreasing the activity of or shutting off P38, resulting in a decrease or shutting off of DUX4. Losmapimod has previously been studied in other indications, which has resulted in a robust experience with safety in over 3,500 participants in over 25 clinical trials worldwide. Based on this experience, losmapimod has demonstrated a tolerable safety profile. We have taken a thoughtful and comprehensive approach to the development of losmapimod for the treatment of FSHD. In addition to our preclinical studies, we have completed several preparatory studies in which we developed, in collaboration with our partner AMRA, a whole body musculoskeletal MRI protocol providing an assessment of muscle health, and a phase 1 study demonstrating tolerability, and that we are achieving concentrations of losmapimod in blood and muscle that, based on our preclinical experience, will result in the reduction of DUX4. In our phase 2 studies, losmapimod demonstrated disease-modifying properties as evidenced by efficacy on structural and functional measures of FSHD disease progression. Today, we will discuss the results of these studies, which have both completed the main treatment portion and are currently in the extension phase. The hypothesis that guides Fulcrum's losmapimod trials is that by decreasing DUX4-driven gene expression and activity, losmapimod will slow or stop muscle cell death and fat accumulation in the muscle. This slowing of FSHD disease progression would be demonstrated by preservation of muscle health, which can be observed on MRI, function assessed with clinical assessments like reachable workspace, and quality of life as reported on validated questionnaires by people living with FSHD. The phase 2 clinical trials are deliberately designed to interrogate a spectrum of relevant disease measures associated with FSHD. Fulcrum worked closely with key opinion leaders and people living with FSHD to design trials that measured clinically meaningful outcomes. A variety of both experimental and established endpoints are included in the trials. Our goals were to increase understanding of DUX4-driven gene expression and how to measure it, continue to build a robust database characterizing safety and tolerability, and ensure that drug exposure and target engagement were consistent with prior trials, as well as our expectations based on preclinical evidence, and to assess structural and functional assessments. The molecular endpoint was DUX4-driven gene expression. From a clinical safety perspective, the trials utilized MRI measures to quantitatively assess fat accumulation and overall muscle health, a number of functional outcomes aimed at characterizing muscle strength and function, as well as patient-reported outcomes to determine the potential for patient-recognized benefit over the course of the trial. REDUX4 is a 48-week randomized controlled study, meaning the participants were assigned to either placebo or losmapimod. REDUX4 enrolled 80 adults ages 18 to 65 with FSHD1 at 17 sites in 4 countries, including the United States, Canada, France, and Spain. 40 participants were randomized to either the losmapimod or a placebo group. The open label study, or OLS, was a study in which all 14 participants were given losmapimod for 52 weeks at a single site. Both studies have ongoing open label extensions in which all participants are taking losmapimod. The average age from both studies was about 45 years old with a slight male predominance, but there were really no differences in race, ethnicity, BMI, or repeats across groups or between studies. It should be noted, however, that in the OLS population, nearly half had a clinical severity score, Ritchie score, of 4.0, suggesting that these OLS participants may have had a bit more severe disease than those in REDUX4. In REDUX4, there were 3 discontinuations in the course of the study, 1 who was receiving losmapimod and 2 receiving placebo. None of the discontinuations were related to study drug. 98% of the participants elected to continue in the open label extension. In the OLS, there were no discontinuations during the study. An extension was offered, and 12 of the 14 participants elected to participate. Losmapimod continued to demonstrate a favorable safety and tolerability profile, with a similar number of participants reporting adverse events during the study. The most common adverse events were dyspepsia or stomach upset, skin-related issues such as rash or dry skin, and increased ALT, which resolved with continued dosing. No participants discontinued or withdrew from the study because of any of these events, and there were no deaths during the studies. There were 3 serious adverse events during the REDUX4 study, but none were related to study drug. Fulcrum, in collaboration with AMRA, developed a new MRI protocol that moves from sampling a slice or slices of muscle to imaging of the whole muscle, proximal to distal, including upper and lower extremities and the trunk. With this protocol, and through quantitative measurements, we can identify a personalized set of muscles at risk of progression to follow over time. We evaluated skeletal muscle quality using a novel whole body MRI methodology, evaluating 18 muscles bilaterally, 36 total, including the rotator cuff muscles as a control, as these muscles are not affected by FSHD. This approach captures the intra and intermuscular heterogeneity of the disease and overcomes the limitations of older MRI measures by allowing for both a holistic and quantitative picture of muscle health. But specifically, Specifically, muscle fat infiltration, or MFI, is an indicator of muscle quality and likely the earliest indicator of muscle disease progression. It is a quantitative measurement of how much fat occupies functional muscle. Based on MFI and other quantitative measures, we are able to categorize muscles into three categories: normal appearing or A muscles, intermediate or B muscles. These are the muscles at high risk of progression and are functional but clearly affected by disease, and C muscles or end stage, and these muscles are so severely fat replaced they have lost most, if not all, function. You can see by the avatars, each representing a different patient from Redux 4, that this whole body MRI protocol captures the intra and inter-patient heterogeneity of the disease. In Redux 4, participants treated with losmapimod showed less muscle fat infiltration than placebo. In the panel on the left, the x-axis shows placebo in blue and losmapimod in orange. The y-axis is change from baseline in percentage points. In the B muscles, those muscles most likely to progress, we observed that placebo-treated participants had approximately 0.5% increase in muscle fat infiltration compared to placebo. In the panel on the right, in the A muscles, or normal appearing muscles, show little or no fat accumulation. Stabilization of fat accumulation was observed in the open label study. Additionally, we observed that there were moderate and strong correlations of MRI with clinical outcome assessments in FSHD. FSHD relevant functional and strength assessments were performed, which included dynamometry, an assessment of upper and lower extremity strength, reachable workspace, an assessment of upper arm and shoulder function, and timed up and go, which is an assessment of patient mobility. Reachable workspace is a 3D motion sensor based outcome assessment that offers significant advantages over traditional technologies because it allows one to quantitatively measure upper extremity movement along 5 different regions. Evaluations are performed with and without weights. Weighted assessments are meant to stress the system and further accentuate functional disability. Regions of reachable workspace are related to activities of daily living, such as washing one's hair or putting away things like dishes in the upper regions of Q1 and Q3 or above the shoulder, or in Q2 and Q4 below the shoulder, putting on pants or eating, and Q5 reaching behind to put something like your wallet or a cell phone in your back pocket. Importantly, it has been reported that the reachable workspace assessment closely correlates with a validated assessment of activities of daily living. On the right, we have the data from Redux 4, and on the left from OLS. In the graphs, on the x-axis is the change in relative surface area, or RSA. The dotted line means no change. Movement to the left of the dotted line is worsening, and to the right is improvement. Assessments were recorded for the non-dominant and dominant arm. Top plots are assessments without weight, and bottom plots are assessments with weight. Again, placebo is in blue, losmapimod is in orange. Losmapimod resulted in the slowing of progression and even improvement on multiple reachable workspace metrics compared to placebo. There are clinically relevant differences in reachable workspace. With weight, there are significant differences in the total surface area in the losmapimod group as compared to placebo. And while the placebo group lost about 2 to 4% of total surface area, the losmapimod group showed trends of slower disease progression, and when using weights, significant improvements of up to 1.5% in surface area, upper body mobility and function, which can be impactful on activities of daily living, such as independent self-care. Similar trends are observed in the OLS study. Now, moving on to dynamometry, again, data from Redux 4 on the right, OLS is on the left. On the x-axis is the percent change, and on the y-axis are the different muscle groups. The dotted line means there is no change. To the right is improvement, and to the left is worsening. Placebo is in blue, losmapimod is in orange. Significant differences between placebo and losmapimod are observed for the right ankle dorsiflexor and non-dominant shoulder abductors. On the right, over 48 weeks, losmapimod showed loss of muscle strength and in some cases, improved strength compared to baseline. The placebo group lost approximately 15% of strength in the shoulder abductors and ankle dorsiflexors, where the losmapimod group showed trends of slower progression or improvements in strength in these muscle groups. These findings in both the proximal and distal muscle groups, in particular those with the shoulder abductors, relate to changes in reachable workspace already noted, and again inform on the progression of dystrophic changes in FSHD. Similar trends were also observed in the OLS study. Now moving to the evaluation of the timed up and go test, here we have on the y-axis the change from baseline in seconds. In Redux 4, we see that the placebo-treated participants showed a trend in increased tug time, representing progression compared to losmapimod. There is an increase of about 3/4 of a second in the placebo group and an increase of less than 1/4 of a second in the losmapimod group, with a difference of 1/2 second. In the OLS study, we see little or no change in tug time. Putting this data together, the structural changes or lack of fat accumulation supports improvements or slowing of disease progression that is observed in FSHD relevant strength and functional assessments. Despite demonstration of a clinical benefit in both studies, technical and biological challenges prohibited observation of change in the DUX4 biomarker in muscle biopsies. In the graph on the right, time is on the x-axis and DUX4 expression is on the y-axis. At baseline, consistent with what is known about FSHD, there is considerable heterogeneity of DUX4 expression, which is likely related to its variable and scarce expression. This variability is noted at all time points. Additionally, there were technical issues related to the procedure itself, which contributed to the difficulty of this assessment. Benefit of treatment with losmapimod was reported by participants in both studies. We asked participants to rate how they were feeling with the Patient Global Impression of Change scale, and they were asked, since the start of the study, my overall status is, and they rated on a 1 to 7 scale, with 1 being very much improved and 7 being very much worse. Looking at Redux 4, on the right is the percent of patients reporting improvement in the placebo or losmapimod group. 4 times as many participants reported feeling improved on placebo, with several participants feeling very much improved in the losmapimod group as compared to none in the placebo group. On the left is the number of participants reported worsening, with 13% of participants in the placebo group reporting worsening, but importantly, approximately 10% reporting being much worse, while no participants in the losmapimod group reported feeling much worse. The totality of evidence supports losmapimod as a potential transformative disease-modifying treatment for FSHD. These results support continued development of losmapimod in FSHD. Fulcrum is committed to the continuing development of losmapimod and bringing access to patients with FSHD as quickly as possible. We understand the urgent need for patients with no or few treatment options to gain access to therapies prior to full regulatory approval. The safety and efficacy of losmapimod is still being investigated according to planned clinical development program. We are encouraged by the results of the phase 2 clinical studies and are planning to meet with regulatory agencies to discuss the design of the next study and the potential path to registration. Successfully completing the necessary studies in the clinical development program for losmapimod is required to support the opportunity for regulatory approval that would allow broad access to people living with FSHD. As we engage with regulatory agencies and understand the data requirements to support registration, we will continue to evaluate the opportunity to provide access to losmapimod outside of registrational clinical trials. Our goal, when possible, is to allow limited access to Fulcrum's investigational products when doing so is safe for the patient and does not disrupt or slow down this development process. Please visit the link provided to learn more about our expanded access policy. In acknowledging the long list of contributors to these results, I would like to first thank the participants of these studies, the people living with FSHD every day, as well as their caretakers and the investigators for the dedication and fortitude to complete this study in the face of a global pandemic. I would also like to thank our expert advisors, the regulators, and our development partners. Thank you for your time, and I'm happy to take any questions.